Hello, my name is Cecile Janssens. I'm a professor of epidemiology at the Rollins School of Public Health at Emory University, Atlanta. And the title of my talk is Predictive and Not, Understanding the Mixed Messages of Our DNA. I'm going to tell in this half hour how it can be that DNA at the same time is predictive for some diseases and not predictive at all for others. I am taking questions at the end of my talk. You can write them down, and so we will go over them later. And please do write them down because I like to answer those. So why we think that genetics, so that DNA is very predictive, is because we are used already for decades to genetic testing for monogenic diseases. Diseases like familial forms of cancer, breast cancer, colorectal cancer, and several dementias, also for Huntington's disease. We know that the genetic mutations play a very strong role in developing those diseases. So I've here simplified it very much. You have a gene and mutations in that gene in some kind of pathway, it's not as simple as I present here, lead to the disease. And that cause of the disease is so strong that, for example, for Huntington disease, if you carry mutations in the Huntington gene, you know for sure that you will develop the disease later in life. You do not know when and how serious, but you know for sure that you're going to get it. Also, when you don't carry the mutations, you're sure that you don't get the disease later on. And why it is so useful to do genetic testing for those diseases is because the risk difference between carrying and not carrying a mutation is so very huge. For the cancers, for example, it makes sense to identify the carriers from the general population and offer them special intervention programs, special surveillance programs, such as we have for the familial forms of cancer. Yet there are also the current interest is also very much on complex diseases, and complex diseases is basically the other end of the spectrum where the genetics is not really a major cause of the disease. It is one risk factor in a whole net of other risk factors, and the environment, I've summarized it here with only one variable in my model, but the environment plays a very large role. And such a large role that each genetic variant, and here you see in this picture here below, you see 3 variants that are implicated in type 2 diabetes, pipergamma, CAPN10, and G7L2. They each play a minor role in the disease, and you see that easily between the risk difference between carriers and non-carriers. And comparing this graph with the one that I presented earlier, you immediately see also why we don't do an effort to, for example, identify the carriers for pipergamma from the general population, because they have only a slightly higher risk of type 2 diabetes than the one who don't carry the risk variants. But the idea then is when we think about the genetic origin of this disease, also genetic prediction of those complex diseases, is how predictive can a test be when we combine in a risk model the effects of pipergamma, CAPN10, G7L2, and all the tens or maybe even hundreds now of other type 2 diabetes genes that we know so far. And that has got a lot of interest in the past years, and that is the topic of my research. So we've become quite optimistic about the prediction of common diseases by DNA, and that is largely due, it's definitely fueled by the enormous discoveries in this field. In genetic research, they have discovered so many susceptibility variants that are implicated in all these complex diseases. And all these discoveries have fueled my field of research. There are numerous studies these days in which the research group try to investigate how predictive common variants are for the prediction of common diseases. Not only genetic risk factors alone, but also in combination with non-genetic risk factors that we have known for long before. And also, we've become quite optimistic because very early on there were already several companies who tried to sell personal genome services via the internet, where using a very simple procedure, and you see that here, where you get a spit test delivered at home, you spit in it and you send it to a lab, and then 6 to 8 weeks later, maybe even faster, you get your predicted risks for tons of diseases at your computer at home. This is a very simple process, and very early on, I think since 2008, these companies were already selling this. But the challenge is, of course, in the following. What happens between the lab test? How do my results get from the lab onto my computer? And here is where genetic prediction plays a role. And that is not as easy as the companies may have suggested earlier. What this company is doing, and here's an example of 23andMe, and I show later a slide, of course, that most of you will know that they are no longer doing this, but in the past they're presenting then your results as really for organized by disease. Here are my diseases, this is my test result, for which diseases I do have an elevated risk, for which I have a decreased risk or an average risk. And what you see then, what they do here is that for the disease, they give me my risk and the average risk and my risk compared to average. And I'm going back shortly, because here you see that for some diseases I really have an increased risk, whereas for others the risk is in absolute numbers also increased, but in relative numbers not so much. Now, what is the shortcoming of these tests is that, for example, for the prediction of coronary heart disease, they only base this prediction on my genetic risk and do not include my non-genetic risk factors, and that is really a shortcoming of this test. There are many more shortcomings, and that is one of the reasons that the FDA quite recently, I think November last year, decided to stop these services from selling their products any longer via the internet. There's not enough data to support that these tests are really very predictive. The FDA says, well, we want you this evidence that it works, and 23andMe says we will provide you the evidence, but we are a bit late. Well, I wrote recently, so in November, a piece about it in the Huffington Post, that this is not really the total story. It is that there is no evidence, and both the FDA and 23andMe dance a bit around that fact. And there is no evidence because it is very difficult to predict common diseases by multiple genetic factors. And here is an example of just one study that investigated this with not too many SNPs, because otherwise it didn't fit on my slides. But this is a typical genetic prediction studies that you will see in the literature. A study had 40,000 individuals where they computed a genetic risk score for all individuals based on 10 single nucleotide polymorphisms. And these single nucleotide polymorphisms, they were selected from GWAS studies earlier on, and sort of this is a bit of a replication study in these 40,000 people. They constructed a regression model, and they did that by entering all the SNPs as separate variables into the model. And here is what you typically see as table 1 or table 2. All the SNPs here on the left column, you see the names of the SNPs. And what I always first am interested is in the odds ratio, namely that's the impact of that specific SNP on the risk of disease. And usually this is an allelic odds ratio. That means that if you have one allele of this first SNP, it increases your risk of disease by 1.13. Now, and to me, this is already enough information. So when this, all these numbers are very, very close to 1, and it's only 10 SNPs, then my gut feeling immediately says that this is not going to be very predictive. And that is then confirmed by graphs like this, that is then typically shown. Here you see a risk distribution, but on the x-axis you see the number of risk alleles for each individual in that study. Some individuals had less than 4 risk alleles on those 10 SNPs, others more than 14. Of course, just to remind you, each SNP you can have 2 risk alleles, one from your father, one from your mother, and so the maximum number of SNPs that the risk alleles that someone can have is 20. Here you see a range from 4 to 14. And what you see here in light gray is the risk distribution for the controls, for the people who did not develop the disease in that study. And in gray, dark gray is the cases. And what is most important in this graph, if you really look at it from a bit of a distance, is that the risk distribution for cases and controls largely overlap. And I will show you later an example where they are more separated. Now, when they largely overlap, it is very difficult to make a distinction between who develops the disease and who doesn't. And that is then also showed typically in an area under the ROC curve. The ROC curve is projecting for each possible threshold that you can make with your risk curve is projecting the true positive rate and the false positive rate. And when a test is not predictive at all, then the area of the ROC curve follows a diagonal line. When a test is perfect, it goes all the way in the corner, so vertically up along the lines. And here you see that for this test, the area under the curve, both in the development data set, so where they made the model, as in the replication set, was only slightly better than tossing a coin. And here you see an example what happens when your prediction model has stronger predictive ability. Here you see a comparable distribution like we saw before, but this is then not for colorectal cancer, but for type 2 diabetes. Again, the area under the curve is 0.60, and when the area under the curve is 0.60, 0.55, distributions largely overlap. Here in the right graph, you see the risk distributions when there are a few predictors that have a stronger impact of disease. And for AMD, age-related macular degeneration, there are a few factors that don't increase the risk by 1.1, 1.15, but really by 3 or 5-fold. And when the risk factors are that strong, you see that the risk distributions of cases and controls becomes more separated. Now, why is that important? Because in practice, if you want to use it for the prevention of age-related macular degeneration, we don't have separate treatment for every score of this model. No, instead we have to make a cutoff score somewhere that we say people on the right side of the line get treatment and people on the left side don't get it. And when your risk model is more separated, when you get a stronger predictor, it's more easy to get all the patients, or most of the patients, the people who will develop the disease on the right side of the line, and all the people who will not develop the disease on the left side. And therefore you need a high AUC. Now, what is a problem with most of these tests these days? We largely see that risk distributions still overlap for most of the risk models that we are making. There are exceptions, like the age-related macular degeneration that I showed before, but also for celiac disease. It's also much stronger. Here you see in the right lower corner, you see an area in the curve how you would like to see it, where it's really going up to that left corner. Now, what is a problem sometimes when people make a risk model and they think it is predictive, that easily, and this is the study that I showed before, that I'm going to show now, that sometimes you see in the news that scientists discover a test for which they found strong predictive ability. Here, this is a test that was recently in the news. There was a press release by the university itself, but when you look at the data of the study, that's what I'm showing here, the genetic risk prediction for alcoholism, where they try to predict alcohol dependence and alcohol abuse in several populations. I have to discuss it only shortly. What they did here, and that is why I emphasized in a previous study that those 10 SNPs for colorectal cancer, they were selected from GWAS. Here we see that they started with 713 SNPs from which they could make a selection to make a risk model. They selected the 66 that were most predictive and made a risk score from that. Well, here again is 2 variants, one from your father, one from your mother. Range gives a theoretical range for the SNP score from 0 to 132, which for ease of interpretation they rescale to 0 to 100. And I say this for a reason, because here are the results. And what you see with the asterisks is that the difference between the controls, the people who were not alcoholic, they were or did not have alcoholism, they are in blue, and the people with alcoholism in red. And what you see with the asterisks is that the ones in red had a significantly higher risk score, SNP score, than the controls. But when you look at the axis on the y-axis, you see that it's totally zoomed in, and it's only a difference of 1 point. Now, that is also where you see here the ROC curve. It looks exactly like the one that we saw before for colorectal cancer. This test, the diagonal means that the test does not predict at all. And here we see that it's only slightly better than a random prediction. I commented on this before too, because I find it very important that when we communicate genetic results to the public, that the public gets a good representation of that, and we should not contribute with our findings and what we want to see to inflated expectations about what we can do with DNA prediction. So the main problem in genetic research is getting the right evidence. So we need to find the right studies about predictability and actionability, but what I showed before is also getting the evidence right. You know, it's when our results, when our studies show that the test is not so very predictive, we have to get clear about that. This is not only for common diseases. We also see that it's very difficult to get the right evidence where it comes to the interpretation of monogenic diseases. And we see this discussion going on in, for example, in whole genome sequencing, where researchers want to sequence a person's entire genome for different reasons, but then you have this discussion that they find a lot of other mutations that are not the ones that they want to find, but that are some what we call incidental findings. And there's a lot of discussion on what we should do with those incidental findings, and the American College of Medical Genetics last year made a list of 56 genes that definitely need to be returned to the patient. It should be informed to the patient, irrespective of their age and whether they want to know it, yes or no. Because the American College find it so important that these factors are, when they are found, then you have to think that, for example, on BRCA1 and 2 for breast cancer. Well, why do I say it in this context? Because also here is a lot of discussion. It is not really very clear when we should communicate something back and when not. And it is because also there's a lot of discussion about the clinical relevance of several mutations. You know, it would be very easy when a mutation has a strong impact on disease risk, like for example BRCA1 and 2, and there are clear interventions that you can do, that you then communicate it back, but the interventions are not always clear. Here you see a table that's from a collaboration that I mentioned earlier from Jonathan Burke. They published it in Genetics and Medicines in 2013. They have a collaboration about the return of incidental findings, and here you see the opinions of 4 participating institutes. Here on the left column you see all kinds of diseases, and in the middle columns you see whether they agree or not agree, or whether they think the mutations in those genes should be communicated to people, yes or no. And then in the right column you see all kind of discussion about it. So also it's not clear for the common diseases what we should, that we have the right evidence yet, but also for the monogenic diseases we still need to discuss a lot. So the basic question when it comes to how predictive is our DNA, whether it's for common diseases or for rare diseases, it always comes down to these 4 simple questions. Whether DNA is predictive depends on what is being predicted. What is the disease? Is it a common disease or a rare disease? In whom? How? So using which genetic variants and for what purpose? That are, when you think about prediction research, the 4 basic questions. And these questions 1, 2, and 3, they together determine the predictive ability. So it is important to understand that DNA does not have predictive ability as such. DNA is not predictive or it is not predictive. It really is depends on what are you testing, which genetic variant, is it a mutation, is it a polymorphism, is it a copy number variations, and for which diseases. So a certain polymorphism may be predictive for one disease but not for another. It also sometimes varies between populations. Does it not hold for Huntington disease, for example, because when you test for Huntington disease in people with a family history or adopted people who do not know the family history, it is very predictive. But we know, for example, for genes that are implicated in obesity and diabetes, that they may have different effects depending on whether people are lean or obese. That's for diabetes, of course. And question number 4 determines whether the predictive ability is high enough. And it's also important to realize there's not so much to do with the predictive ability, but for example, when you think about the prediction of breast cancer based on single nucleotide polymorphisms, that might be predictive enough to stratify a mammography screening program, but it might not be predictive enough to recommend a specific woman whether she should take prophylactic surgery, yes or no. So a certain risk score might be predictive for one goal, but not for another. And this is, and I'm going to discuss 2 factors that are very important in the predictive ability of common diseases. And one reason why the predictive ability of common diseases is not very high is that most complex diseases are only partially heritable. And here you see on the right side, you see a list with heritability estimates, that the degree to which the variation in the development of the disease is attributable to genetic factors. You see a list of diseases and traits that vary from a very high heritability to a very low heritability. And what does that mean? It means that even if we totally understand the genetic background of diseases, we uncovered all the variants that are implicated, including the interactions, etc. It means that if you use a DNA test to predict those diseases, you likely will be very successful in predicting eye color and type 1 diabetes, the diseases and traits on the top, but very, very unsuccessful in happiness, lung cancer, type 2 diabetes, and the non-familial breast cancer, simply by the fact that non-genetic risk factors have such a big impact on the disease risk. And that is illustrated here by the picture. At the left here is identical twins who evidently do not have the same risk for type 2 diabetes. Another reason why complex diseases are so difficult to predict is because there are many, many, many causal combinations that lead to the development of the disease. And interestingly enough, we immediately understand that when it happens to disasters, which are also complex events, but we tend to be over-optimistic when we try to imagine the predictive ability for common diseases. So here you see 4 ships that capsized and 4 planes that crashed, and I think we all understand that these did not crash for the same reason, but also not for a single reason. And that's the same in complex diseases. An example that has been used in disaster management, and which I'm presenting already for quite some years, definitely since 2008, because it's so illustrative of how I think that complex diseases are finding their cause. So this is the Herald of Free Enterprise that capsized in 1987 in front of the coast of Belgium, and it became a case study in disaster management because the ship capsized on a very clear day. It was a good ship, there was nothing wrong with the staff, everything seemed in perfect condition, and then 100 meters from the coast it capsized, killing almost 200 people. And they analyzed that, and they found out that there were multiple factors together that happened approximately simultaneously that together caused the capsizing of the ship. And there are some factors that are more obvious than the others that I showed on the next slide, but the bow doors at the back of the ship were open. And that is, you see it on the left graph, you see it on the back of the ship in the red part. And that's what they also had was that the ballast tanks were full when they departed. You have to understand that ballast tanks you need to fill with water when it's high tide, and you need to get cars on the deck of your ship. You have to lower the ship first, and you do that by entering water in the ballast tanks. But then before you sail off, you need to empty those ballast tanks. Also, they were delayed at that day, and so they were sailing with a higher speed than normal, and what the higher speed does with the ship is makes higher waves. And you can feel already what was going on. These higher waves could enter the ship because the bow doors were open and because of the full ballast tanks. And so the ship was low on the water with open bow doors, the higher waves could enter the ship. And because the cars were not properly secured on the deck, they started to move when the water came in and the ship capsized. Now, this is how they summarize that in disaster management. They make a why-because graph, and they analyze, they break every cause, they break into separate causes until it can no further be broken down. And they concluded that the Herald of Free Enterprise capsized because of a combination of 13 risk factors. And this is also how common diseases might occur. It's a combination of many, many risk factors that in each of us occur in a different combination. So it's very difficult, also for the prediction of common diseases, but also for common disasters, it's very difficult to prevent common disasters when the causal mechanism is every time different. So the only thing you can do with this information is analyze a lot of these capsizes and then count the number of risk factors and intervene on the most important risk factors. And that is, of course, what we are doing with many common diseases in public health. We intervene on body weight, on smoking, on exercise, on diet, etc. And so, because the common diseases are very complex in their origin and only partially heritable, they may not be so very predictive. And here in this graph we summarize the predictive ability of DNA. So the prediction by DNA depends, the predictive ability depends on the heritability of the disease and by the genetic complexity. So diseases in the upper left corner that are very heritable and genetically simple because it's only one mutation or a bunch of mutations in one gene, when we know these genetic origins, we can do very good prediction of disease. And these diseases we know as monogenic diseases, which have a very simple cause. On the lower end of the graph, the whole red area, is the area where the heritability is low. And when the heritability is low, for example in lung cancer, the non-familial breast cancer, and type 2 diabetes, then predicting diseases based on a genetic test only will never be very predictive because there are too many other factors that impact the disease risk that are not considered in the test. Then on the right corner of that graph, the upper right corner, is the diseases that are and traits that are very heritable, but that are genetically in their origin so complex that it's also very difficult to predict those, not because they're not genetic enough, but that in order to make good prediction, you need to construct a good model. And when it's genetically too complex, it will be very difficult to make a model that is so precise that it can ultimately predict very well in individuals. So this is then the limitations to the prediction of common diseases. Now, will that get better with all the new developments that are going on? Will we get better prediction of disease when we sequence the whole genome instead of looking only at common variants? Will it get better when we do larger studies? Our study is not large enough, is that a problem? Will we get better prediction when we make more complex models instead of a simple logistic regression model? And will it get better prediction when we do not look only at DNA, but all kind of other omics parameters as well? Well, here is my short answer to that. The question for whether these new trends can make the prediction better really depends on, can they increase the heritability or reduce the genetic complexity? Whole genome sequencing may explain a larger part of the heritability. We may discover more variants that are implicated, but it does not increase the heritability of the diseases. The same is for more complex models. Yes, when we do it more sophisticated, maybe we explain a better part of the heritability, but it will not make the heritability higher. And it also not reduce the complexity. And large studies is the same thing with whole genome sequencing. We might discover more variants, but the question is they don't change these two parameters that are in the graph. The only thing that does change the parameters in the graph is considering other omics parameters like metabolomics or but also imaging parameters. And why that does change it is because it may improve the prediction, but then we are no longer looking at genetic prediction only. We are taking many other variables in the model. And so you can expand this graph in this way because heritability is just a measure of explained variance. And when your variance gets bigger because you take other explanatory variables into the model, you can move up in this graph and maybe get into the green zone. So it's not very obvious that these new developments immediately improve the prediction. And that is because the prediction ultimately is determined by the diseases that we are going to predict or the traits that we are going to predict, and not so much by all the technology. So here is to summarize my talk. So we have on the one side, if we consider this a line from perfect prediction to very poor prediction, on one side of the line are the monogenic diseases that we can predict very well. On the other side, and I take a very broad spectrum for that, is the common diseases that are never get very good in prediction. Then to finalize my talk, so what can we do, what can be done using DNA? What can we predict? Here is a list of outcomes that we would like to predict with using DNA, and here's my summary of how good we can predict them. The rare monogenic diseases, yes, we can predict them very well. And the hereditary disorders, the syndromes, we can predict them very well because they are genetically very simple. The common diseases, I'm not so very optimistic. Pharmacogenetics I leave in the middle because there are some variants with strong effect and others are smaller effects. The outbreaks and the characterization of bacteria, there's increasing research about that. It's very useful to characterize bacteria and see how an epidemic spreads across a region. Very useful applications there. Forensics, it depends a bit what you're doing with it. It's very useful for matching. When you find DNA on the crime scene and you have a database with suspects, you can match whether that DNA is one of the suspects. But when it comes to prediction of eye color, hair color, and length, etc., it's not so very predictive. And finally, nutrigenomics, that is too complex to discuss. So when we then look at this overview, here is the two things that we can do with it. When it comes to diagnosis and matching, DNA is very predictive. But when it comes to, very informative, let me say this, but when it comes to prediction, it's not so informative at all. And then here is my final slides to conclude this talk that I recently summarized in Wired. The predictive ability of DNA is limited not by technology but by the complexity of the causes and the heritability of the diseases and traits. DNA testing might not predict diseases very well, but it can diagnose them. It will not be able to predict who's going to commit a crime, but it might help find out who did it. And it's not going to predict who will be the best father for a child, but it can help find out who the father is. DNA might teach us more about the past and the present than about the future. That was my talk. I'm now going to see some questions that are raised in by the participants. And so one of the questions that was asked was about the commercial companies, and the question was, is it fine to go by the test reports given by any personal genome company in the present scenario? And then I cannot, as in Europe, many companies are still running. It depends, of course, on what they are testing, but when it is testing for common diseases, I would definitely say no, because we can't predict those diseases quite well, and most of all, we also have no no evidence. You always have to look for what DNA variants are they exactly testing. But so far, to the best of my knowledge, these are not predictive enough. We have recently published a paper. I did not discuss it in this talk. It's by Kalf et al. And also on my name. You can search it in Genetics and Medicine, where we compare the predictive ability of 23andMe, Navigenics, and DecodeMe. And I think this paper, you know, it's only 3 companies, but I take them very representative for all the others that are out there. So I would never ever take a medical decision based on that. Are the traditional clinical analysis considered as a basic predictor for diseases before to go to the next step with DNA diagnosis? That question was asked by Maria Dolores Sotelo. I do not know. The clinical genetics is not my field of expertise, and I do not know enough to give a good answer on this. Are all hospitals with programs that support DNA diagnostics for patients? Is homosexuality considered for DNA diagnosis? Okay, that's an interesting question. I would not know why you would like to do it, but definitely homosexuality is not that genetic that there is a test that we can use it for prediction. And not even close, I would say. It was in my list, but it was a list of diseases and traits, and not all the traits in the list were diseases. But it is like alcoholism and like also about suicide risk, that was recently in the news. Those traits are so complex, there are so many factors involved that we can never diagnose that or predict that reliably based on DNA. Hospitals with programs that support DNA diagnosis, they are mostly indeed for patients. You need to have an indication generally to get into there. Is personalized genomics a good approach for clinicians to consider? That is a question by Arun Kiran Patnam, and I would like to challenge you to refine what you consider with personalized genomics, because that is a very, very broad term. If you think about the personal genome services like those offered by 23andMe, I would definitely not do it. And I think for everything that is personalized and genomics these days, we do not have yet sufficient evidence. Would I use DNA to predict sports performance? I can give a cynical answer here to Anna Sandoval. I can also give my true answer, no. And my cynical answer is, if you want to be fooled, yes, but that's not a good answer. No, also sport performance, those traits depend so much on so many other variables that it's very difficult to use a DNA test to even say whether you are more likely to become a sprinter or a endurance sporter. I think, you know, these kind of tests for sport performance, for homosexuality, for suicide risk, for alcoholism, it has all been in the media, but there is an enormously wide difference between what is reported in the media and what we find evidence in science. And I think with every trait or every disease that has many, many risk factors involved, it is very unlikely that a DNA test will be a very good predictor. And with that, I answered my last question, and I thank you all for your attendance.